The topic of this webinar segment is lessons learned from a phase 1 trial of heart-derived stem cells as we move into phase 2. And for us, the objective of using stem cells is to achieve true regeneration in the injured heart. When we reflect on what that means, the common definition of regeneration, in this case from the American Heritage Dictionary, is regrowth of lost or destroyed parts or organs. So we actually seek to regrow a part of a tissue or an organ that's been lost or destroyed as a process, as part of the disease process. And if one reflects on the criteria that we might use to assess regeneration clinically, criterion number 1 would be evidence for growth of new, healthy heart tissue. And if it's healthy heart tissue, it should be functional, and therefore there should be improvement of function, at least in the region that has received the cells and where regeneration has taken place. Even though reduction of scar is a clinically desirable endpoint, it's really a bonus with regard to regeneration, because scar per se need not be reduced in the process of regeneration. It is not part of the definition of regrowth of lost or destroyed parts or tissues, but nevertheless, reduction of scar, and in particular the conversion of scar to living myocardium, would be a very desirable therapeutic principle. So if we reflect on the types of stem cells one might use for therapeutics, there are the pluripotent cells, which are not yet, by and large, ready for application, particularly in the heart, where issues such as arrhythmias are going to limit the application of these cells for some time. The whole category of adult stem cells, however, is relatively mature within the heart field. Skeletal myoblasts have been used, bone marrow-derived stem cells have been used. Our focus has been on cardiac-derived cells, and in particular on a type of cell that we call the cardiosphere-derived cell, or the CDC. And in this publication, methods of which are summarized in the present slide, we depict how we get from a human being to a clinical dose. So patients undergo a percutaneous endomyocardial biopsy. It's a catheter-based procedure that's minimally invasive and done under local anesthesia. About 200 milligrams of heart tissue are taken out by remotely activated forceps. These are brought to the laboratory where they're plated on fibronectin-coated dishes and allowed to form a lawn of cells that spontaneously is shed by the biopsies. This lawn is allowed to become confluent and then harvested, and when put through a fax machine, the cells that come out from the biopsies, the so-called cardiac outgrowth cells, express a distinctive antigenic profile, which I'll discuss more in depth later, but interestingly, they self-assemble into three-dimensional microtissues called cardiospheres when placed into culture under conditions that repel adhesion, such as in culture dishes coated with polylysine. These are structures that are 50 to over 200 microns in size, as you can see here, and these would be difficult to apply down the favored intracoronary route where safety has been extensively demonstrated by other methods, namely the intracoronary delivery route, because they're too large and may plug arterioles and cause damage. We therefore derived a dispersed single-cell product by replating the cardiospheres under adherent conditions to yield the cell product, the cardiosphere-derived cell of the CDC, after 2 passages shown here. And in the CADUCEUS trial experience, we achieved yields of about 30 million CDCs within 3 to 5 weeks, and the target dose was 25 million. So you can see that on average, from time of tissue harvesting to a therapeutic option would be within the time range depicted here. CDCs have a number of properties that have been extensively studied in a number of references which are listed at the bottom here, but summarized in the present slide. They are CD105 positive cells that are negative for hematologic markers such as CD45, and they're of intrinsic cardiac origin, as shown by forensic methods in the publication cited here. Less than about 25% on average are positive for CD90, which is a fairly ubiquitous marker of mesenchymal stem cells, and only a very small minority are positive for markers of fibroblasts or myofibroblasts. They're also not mature cardiomyocytes or on their way to becoming mature cardiomyocytes, as they are negative for all markers associated with contractile proteins. On a therapeutic basis, they can be administered to injured hearts where they shrink, scar, and increase myocardial viability. They actually increase the amount of living heart muscle, as I'll show you soon. And when compared to other cells that are in the clinic, human cells that have been used for targeting heart disease, we find that they're functionally superior when applied side by side in the same SCID mouse model with the same endpoints of scar, function, and paracrine ability. The cells fulfill the criteria for progenitor cells in that they're multipotent and clonogenic, but paradoxically, long-term engraftment and differentiation are absolutely not required for benefit. Therefore, we believe that they're not functioning via stemness and the traditional engraftment, proliferation, and differentiation paradigm, but rather through the secretion of factors and interaction with host tissue that then leads to durable benefits that do not arise from proliferation of the engrafted cells themselves. And finally, CDCs are fairly large cells. They're about 20 microns in diameter, which compares to the typical diameter of a capillary at 8 microns, so that they are microembolic. They will reside by preference within the capillary space or even in the precapillary space. A few of them may go through, but retention is expected, and therefore microembolic possibility occurs, and there is a maximum safe dose, which we have estimated from preclinical models in the porcine post-MI model to be 350,000 CDCs per kilogram per artery, which translates to our clinical dose in humans, maximum safe dose of 25 million. So using these sorts of data as background, we performed the CADUCEUS trial. CADUCEUS is the ubiquitous symbol of medicine, and in this case stands for cardiosphere-derived autologous stem cells to reverse ventricular dysfunction. The primary 6-month endpoints were published last year in the publication indicated here. In order to become eligible for enrollment in CADUCEUS, you had to be a patient who had had a prior myocardial infarction within 30 days of screening and have persistent left ventricular dysfunction with an ejection fraction of less than 45%. By the time patients were treated, they were 45 to 90 days post-MI. The study, after qualification, was fully randomized, and the randomization was either to control, which was a conventional therapy, no placebo, or to receive CDCs, either in a dose escalation manner. 4 patients received 12.5 million CDCs. 1 patient received an intermediate dose of 17.3 million CDCs, and 12 received a dose of 25 million CDCs down the infarct-related artery. This is a phase 1 first-in-human study, so the primary objectives were to measure safety parameters, but we also, of course, looked for preliminary indications of bioactivity and efficacy by magnetic resonance imaging. It turns out that contrast-enhanced magnetic resonance imaging is a good way of distinguishing living heart muscle from scarred heart muscle, and also for measuring regional and global function, and therefore gave us all of the parameters that we wanted to measure in order to test the idea that therapeutic regeneration might actually be occurring. Patients who were randomized to receive CDCs underwent intracoronary infusion of autologous CDCs once the dose was ready. There were no safety concerns that arose from the initial analysis of the data or from the final one-year analysis, which has been recently presented and is now submitted for publication. But in particular, there's no significant increase in arrhythmia or sudden cardiac death. This is the first therapeutic modality that both decreases scar and notably increases viable tissue. And the kind of evidence that we use to support that claim is shown here. These are magnetic resonance images from patients who were either randomized, in the case of the top two images, to receive CDCs, or in the case of the bottom two images, to state-of-the-art therapy controls. And what is shown here are cross-sectional images through the left ventricle, which is this donut-like structure, and the right ventricle. Under the settings shown here, living heart muscle appears densely black, and scar appears white. And you can see that within the wall of the left ventricle here, as shown by the cursor, there's large segments of white or scarred muscle that in its greatest extreme occupy almost the full thickness of the anterior wall of the left ventricle, and which span about a third to a half of the circumference of the left ventricle. So a fairly large anteroseptal myocardial infarction. This image was obtained 88 days after the infarction and 2 days before treatment with autologous CDCs. The same subject was imaged 6 months later, and you can see here now that the scar has markedly decreased in thickness and in span or circumference. And much of the scar has apparently been replaced by black muscle or viable tissue, as you can see. And so from these kinds of images, which we can reconstruct volumetrically, we can gain estimates or actually precise measurements of viable myocardial mass and scarred myocardial mass. And from these sorts of measurements, we can get the data that I'm going to show you for pooled results in the next figure. Contrast those findings with these in a subject who had been randomized to receive no CDCs. This patient had a large septal myocardial infarction, and you can see much of the septum between the left ventricle and the right ventricle is occupied by scar, but there's no reduction in that amount of scar over the subsequent 6 months, nor is there any apparent increase in the amount of viable tissue within the zone of injury. So when we look at the pooled data, first for controls, and we quantify the change in scar mass from baseline being 45 to 90 days post-MI, and 6 months later or 12 months later, we see that on average, scar mass does not change spontaneously in subjects who are treated conventionally, nor is there any spontaneous increase in the amount of myocardial mass. These data confirm the traditional pessimistic dogma that scar once formed in the heart does not resolve on its own, and that living heart muscle once lost does not spontaneously regrow. When we look at the results from the CDC-treated subjects in terms of scar mass, the data are quite different. So that at 12 months, for example, we have a reduction of scar mass amounting to about 12 grams, much greater than the insignificant changes seen in control subjects. And this in itself is a desirable therapeutic principle, but not necessarily indicative of regeneration. However, the changes in viable mass were notable, were even greater at 1 year post-therapy. Myocardial viable mass increased by 22 grams on average in the subjects that had received CDCs. The fact that it increases at all is notable and is consistent with therapeutic regeneration. The fact that it increases by 22 grams is particularly notable because it accounts for about half of the initially lost myocardium being regrown over that 1-year interval. 22 grams is equivalent to the packed volume of about 500 million adult cardiomyocytes, and these patients have lost on average about 1 billion cardiomyocytes in the course of their myocardial infarction. So this is evidence for scar reduction and evidence for therapeutic regeneration or increase in viable myocardium. If indeed there's a causal relationship between the decrease in scar mass and the increase in viable mass, as one might expect if one were being converted to the other, we found a relationship between the amount of scar reduction, which is on the y-axis, and the increase in scar mass and the increase in viable mass, which is on the x-axis. That is, patients who had a greater reduction in scar mass also had a greater increase in viable mass. And the function in the region of the heart where the treatment had occurred is improved both by measurement of peak strain using an MRI method that looks at tagging of the segments, or by conventional thickening of the wall, which is a model-independent way of looking at how well regionally the myocardium is working. And in the regions of treatment, actually, the function of the wall increased to control levels. We concluded CADUCEUS and were gratified by the lack of apparent safety concerns, and even more tantalized by the evidence for bioactivity and the indications of therapeutic regeneration. Therapeutic regeneration is something that had never been convincingly shown before in any controlled trial of cell therapy. So we were very gratified by that, but we realized in the course of executing CADUCEUS that autologous therapy is, even though it's viable, is somewhat tedious. First of all, there are timing constraints. It takes 3 to 5 weeks to generate a suitable dose for CDCs using typical myocardial biopsy volumes from tissue harvesting. It's expensive because every patient requires an individual manufacturing run and individualized quality controls. There's risks associated with the harvesting procedure itself, and there's the even greater risk of undergoing a harvesting procedure and then being unable to receive cells due to manufacturing failure. The rate of manufacturing failure in CADUCEUS was less than 20%, but nevertheless, even one manufacturing failure means that a human being has been exposed to tissue harvesting without any potential benefit. Quality assurance and quality control is suboptimal in the sense that one is racing the clock to get the dose ready. It's not like having a master cell bank where you have the luxury of time to do every test under the sun and repeat it several times in order to ensure the quality of the substance being infused. There's inevitable interpatient variability in cell potency because of the comorbidities associated with each patient. So for these reasons, we questioned whether allogeneic therapy or universal donor therapy might be safe and effective with CDCs. We first tested this in immunologically mismatched rats that are believed to be immunologically more different than any two human beings. And we compared perfectly matched cells that are syngeneic to those that, and you can see the color code for syngeneic here and for allogeneic, syngeneic being green and allogeneic being blue. The functional changes over a span of as long as 6 months after the cell treatment of myocardial infarction were the same for syngeneic and allogeneic cells. Focus here on ejection fraction for the same. As an example, you can see it increases markedly in the cell-treated cohorts, whether they're syngeneic or allogeneic. Animals exposed to xenogenic cells, in this case human cells injected into immunocompetent rats, have no benefit, and the controls actually have progressive deterioration of function over the first 3 weeks, which lasts out to 6 months. So in this study, which was published in Circulation in 2012, we actually also demonstrated that there was no development of donor-specific antibodies, nor was there anything but mild histologic inflammation. So we ended up concluding from this rodent study that allogeneic therapy might be viable in the population under study here. We then sought to do IND-enabling studies by doing the same kinds of experiments, but now in a clinically realistic model mimicking the clinical delivery situation. So these are mini pigs that were subjected to myocardial infarction and allowed to heal for 2 weeks, and then at that point were assigned either to infusion of placebo or allogeneic CDCs. You can see here an example of an MRI in a CDC-treated animal before cell therapy and 2 months afterwards. And you can see at a glance that there is less scar at 2 months than at baseline and more viable myocardium, and there's been no progressive remodeling of the left ventricular chamber. When you look at the comparison between controls, placebo controls, and CDC-treated animals, there's a reduction of scar size in the CDC-treated animals that's not present in the controls, an increase in viable mass that's not present in the controls, and preservation of ejection fraction, whereas the controls see progressive deterioration in the ejection fraction over time. In these animals, we also failed to see any evidence of marked inflammation, nor did they develop any panel-reactive antibodies. So, in summary, we find that autologous CDCs produce therapeutic regeneration in humans post-MI in the CADUCEUS trial. And in the preclinical data that I've shown you from rats and from pigs, allogeneic CDCs without immunosuppression appear to be safe and promote cardiac regeneration while improving heart function. We conclude that the improvement of heart function must be by stimulation of endogenous repair, because even though the functional benefits and structural benefits are apparent for months after the procedure, we can find no evidence of persistence of the injected allogeneic cells beyond about 4 weeks after they're introduced into these immunocompetent hosts. So altogether, the work motivates the testing of allogeneic human CDCs as a potential off-the-shelf product for cellular myoplasty, and this is currently actually being done in the All-Star trial. The patient population enrolled in All-Star is somewhat similar to that in CADUCEUS. These are post-MI patients who have an ejection fraction of less than 50%, and additionally are documented to have, by screening magnetic resonance imaging, infarct size involving greater than 15% of the left ventricle. The CADUCEUS population was in the range of 30 to 90 days post-MI, or the healing MI. These patients are being studied in All-Star, but All-Star also additionally looks at patients who are greater than 90 days and up to 1 year post-MI or more chronic MI to see if they have a similar beneficial response. It's phase 1 leading to phase 2 study. Phase 2 will involve 260 patients, in phase 1 14, for a total of 274 patients. Presently, the trials in phase 1, 4 low-dose patients have been treated and 1 high-dose patient. After the full complement of 14 open-label patients has been studied in phase 1 and DSMB approves it, we'll proceed to a double-blind, randomized, and placebo-controlled aspect of the trial, which will involve 20 sites in the United States. This is a study in which phase 1 is funded by the NIH, and the phase 2 component is funded by CIRM. These are CIRM grants to the biotechnology company Capricor. The national PIs are Tim Henry of the Minneapolis Heart Institute and Raj Makkar of the Cedars-Sinai Heart Institute. Patients who are randomized to receive cells will receive allogeneic CDCs without immunosuppression with intracoronary infusion, and the placebo subjects will just have the same procedure but with placebo, and the code will be broken only at the end of the study. So in conclusion, I'd like to acknowledge the collaboration of a number of key individuals in the studies I've described. Raj Makar was my principal clinical and interventional collaborator in CADUCEUS. Kostas Maliaras did much of the pig work with the collaboration of many of the individuals listed here. Capricor collaborated in the allogeneic mini-pig work. And funding for CADUCEUS was from NIH. For the allogeneic pig work was done under a disease team 1 funded by CIRM, which actually was the first disease team to result in validation of a therapeutic candidate and a new IND, which led to the All-Star trial, and some facilities support from philanthropy from the Board of Governors of Cedars-Sinai Medical Center. Thank you.